Swayam Prabha. Digital India. Educated India.
So this is only just to show you that so these are the ligands in biology. So the ligands that I talked about is in laboratory or in the nature, which is precious gemstones that I showed. They contain aluminosilicates and various different kinds of ligands are present with a small amount of chromium, because chromium 3 is colored. So now if you keep changing the group around the metal, color also, the intensity and the nature of the color also changes from red to green. You can change because you bring about change in the energy level between the ground state and the excited state. So the more the energy, the difference, it will have a different color, because the energy required from year to year is different depending on where in what region it appears. So that color is what what is observed is not what is seen. It is the complementary color is what is seen. Okay. So this is just only to show you that metal ion, magnesium and cobalt. So these are surrounded by the ligand. So these are bioligands. These are not the ligands that I wrote here and showed you. That in the laboratory, these are the compounds that are present, but in biology, in our body or the plant cells, they will have all this. Okay. So vitamin B12, chlorophyll, this is the one present in the plants, and the color of the the green of the plants is due to this chlorophyll. Okay. So this is just to show you that these are all these are also, you can call it metal complexes. So metal complexes in biology. So various different kinds of things, reduction of nitrogen to ammonia is done by the enzyme nitrogenase, and that converts nitrogen to ammonia, and that's how the proteins are synthesized by the plants. Or conversion of carbon dioxide to carbohydrate and releasing oxygen, which is what is photosynthesis. All that is done by the metal. So many of these things also, what is shown here, for example, DNA, cleavage, making or breaking of DNA. If the DNA is broken, it has to be repaired, or a new DNA has to be synthesized. That also requires a metal. So in these cases, it's magnesium and zinc. It is normally what is utilized, but in these cases, it utilizes transition metals. So this is where is the hemoglobin and myoglobin. So iron is in the +2 state and it gets bonded to the metal ion. So oxygen is bonded, and this oxygen is what is transported through the blood. So it has which these are all present in the red blood cells, and hemoglobin is a protein. So this green color or orange color or purple color indicates different protein. So it's a tetramer. So one ion is present here, another iron here, third and fourth. So 4 ions and 4 different kinds of proteins are there, and so it is a tetramer. And this oxygen present in each one of these sites is what is transported. This more of a biology. I am sure some of you probably know already.
So what's the difference between the alpha and beta chains?
The way that hydrogen bonding is formed. So it is the shape of the.
So what's the difference between like the two?
No, alpha, the hydrogen bonding, if you put alpha, you know that how the hydrogen bonding is formed. So it depends on the how these one protein and the other protein is arranged with each other. That gives rise to alpha and beta. There is structural difference is there. So that means even the way that these proteins are folded also is important for them for the function. Without folding there is no function. So these whatever I showed you, these proteins have a particular number of amino acids. So these amino acids also there is a pattern. If that pattern is not maintained, then it leads to a problem. One of the two problems of many is what is shown here is what is called sickle cell anemia or thalassemia. So that means the DNA in the protein, what you see is a red blood cell. If there is a change in the structure of the protein, one amino acid is different, is not is changed from a particular amino acid to another one, then this red blood cell will not be of this shape, but it will be instead in this shape. So this is in the shape of a sickle, and hence it is called a sickle cell anemia, because this is not as efficient as this one to transport oxygen. So that is because of the change in the one of the amino acids. There are about 60 amino acids in each one of these chain that I have shown you. So there are about 60 into 4 that many amino acids that are present. So that means among the 60 amino acids there is a particular sequence of amino acid. In that sequence, if one of them is changed, then it will lead to there is a particular one amino acid that leads to this kind of a disease called sickle cell anemia. And there are many people who are suffering from those kind of this is one of the anemia. That means it doesn't have the same capacity to transport oxygen efficiently like the normal hemoglobin. And these the alpha chain or beta chain that I am talking about here, if you look at this one, this alpha, that within the 3 months of conceiving, this alpha is completely formed. The baby within 3 months alpha is completely formed, but the beta gets completed after 6 months. So that means after the 6 months of the birth, so that means complete formation of beta, and then alpha is found within the 3 months. So it's very important whether it is efficiently forming or not. If it is inefficiently formed, then leads to whatever the thalassemia. So thalassemia is due to the incomplete formation of the alpha and the beta. So that means it has to be a proper formation of that chain that I showed you. If it is not properly formed, then it leads to one of these diseases called. So that means even that complete formation are also important. So that efficiency is determined by the complete formation of alpha and beta. Any incomplete formation leads to disease called thalassemia. So in such people who have this kind of a disease, you have to transfuse blood every 6 months once. Otherwise, it will not be efficiently be transport of oxygen. Then they will be anemic. That means they will be weak. So this is what I showed you, iron sulfur cluster, which is present in the spinach. That is the cartoon that I was referring to. So these are very rich in iron, and these are bonded by the. So this is one of the coordination compounds you can call it, but it is present in biomolecules. So hence it is a yeah.
So what does the cysteine do?
Huh?
What does the cysteine do?
What is it?
Cysteine. What does cysteine do?
Cysteine is an amino acid. Otherwise, cysteine is like a ligand here. Ethylene diamine, ammonia. Cysteine is an amino acid. right? It contains sulfur, contains carboxylate. So it's a ligand like ethylene diamine, what I showed you here, or what I showed you is EDTA. It contains nitrogen and then carboxylate. So they bind the, so you cannot have a free ion. You had some question.
So then in this case, is sulfur not the ligand?
Sulfur?
Is sulfur not the ligand in this case because cysteine was considered the ligand?
No, it has both.
Okay.
It has inorganic sulfur as well as cysteine. Both it has. See here, the one that has no connection, that is inorganic sulfur. The one that has a connection, that is the cysteine amino acid. Cysteine has a sulfur, so that functions as a ligand. Yeah, photosynthesis, that how carbon dioxide and water combine to give glucose and oxygen. That oxygen that we breathe every day is coming from this photosynthesis, right? So plants take carbon dioxide and then give us carbohydrate plus oxygen. And the chlorophyll is, this molecule does all that, whatever the energy that is necessary, it captures light from the sunlight, and that energy, whatever necessary to combine carbon dioxide and water to give you carbohydrate, is done by the chlorophyll. So here, basically, if you look at the reaction, carbon dioxide plus water giving you carbohydrate plus oxygen. So there is a reduction and oxidation that is happening. So oxidation of water, reduction of carbon dioxide to make you carbohydrate. So that means electrons are involved, and water and carbon dioxide through the sunlight, water is oxidized and 4 electrons are given and oxygen is released. Those 4 electrons are used to make the carbon dioxide converted into carbohydrate combining with water. Nitrogen fixation is what I talked about, Haber process. In nature, this is the method that nature uses to convert, make ammonia or nitrogen containing compounds. How proteins or amino acids are made? The plants have the capacity to convert nitrogen into ammonia. So that is what is called nitrogen fixation. Why the word fixation? Because nitrogen is floating around us, but you have to hold it, fix it. That means make it react. That's what is the fixing here means. So nitrogen plus electrons are required and also protons are required to make ammonia. So this is done by the iron molybdenum clusters and then ion clusters. So otherwise it is not very a simple reaction. Haber process uses very high temperature and very high pressure, 400 degrees Celsius and then very high pressure. But nature does it at room temperature, right? So half of the nitrogen containing compounds that we eat every day are coming from plants, the other half coming from Haber process. So replication, repair and recombination of the DNA, this happens every day in our body, and that is carried out by the magnesium and then zinc containing molecules. So metals in medicines are also, so these are all ones that are introduced from outside. So metals in medicine, so various different kinds of metals are there that have applications. You can see here gallium, gadolinium is something that I was talking about, used for magnetic resonance imaging. So cisplatin, carboplatin is one of a modification of the cisplatin. So various different kinds of compounds are there containing metal. So lithium, gallium, technetium, bismuth, barium, gold, gadolinium and then samarium. So these are all different kinds of compounds containing metals that are used for diagnosis or for treatment. So simple molecule like here what is given transplatin is cisplatin. Cisplatin is used for treating different kinds of cancers, but at the same time transplatin is not, transplatin is toxic. So you can see the difference between the two compounds, the position of the with respect to ammonia and chloride. If the ammonia is here, then you have a transplatin, and that transplatin is toxic, whereas cisplatin is, it cures some of the cancer. So different kinds of simple compounds is what is shown here. Gold compound with carbohydrate, what is shown here is used in arthritis. Just to give you a few examples, such simple metal compounds have lot of applications in day-to-day life. So very few selected examples I have shown here, but there are many. So this is the cisplatin. So basically cisplatin, what it does, this is the DNA. So in the body where you have water, the chlorides can be easily replaced. That how chlorides can be easily replaced also coming from the crystal field theory that hopefully you will be learning in your 12th class. Otherwise, if you take iodine here, I minus and I minus iodide, then that will not be a good ligand. That will not be good for replacing the iodine by water, because the soft nature makes it very strong bond and it will not easily get replaced. And also there is other reason why what is called strong field and weak field ligand. So chloride is a weak field ligand. So weak field ligand means easily you can remove it. Strong field means you cannot remove it. So how do I say it is a weak field ligand and strong field ligand? That comes from crystal field theory, which I did not talk about. That's something that you probably reading in your 12th class. Okay. So metals are also toxic, particularly mercury. In the olden days, mercury was used as for various different kinds of as electrodes and everywhere, right? Battery, even today, some of these compounds are used, but slowly they are replaced by other non-toxic compounds or materials. So they are highly toxic because mercury easily gets vaporized, and vaporized means you breathe, and if you breathe, it will get it will attack the amino acids and proteins which are soft in nature. For example, cysteine contains a sulfur ligand. So mercury and sulfur easily form a bond, and cysteine will not be able to do its function because mercury has taken that sulfur of the cysteine is taken by the mercury. So cysteine is not available. Just an example to give you, that is something that I showed you before. One of you asked the question, why cysteine? So soft interaction will make it a strong bond, and now cysteine, what is necessary for the function, is not available. Just one example to give you. So many different people have contributed to Alfred Werner is the one to begin with, but there are many various, these are all modern people who have come up with many different kinds of things to understand the chemistry. And the one slide I happen to have, these are the guy, is the guy Ramsay, who was the first director of the Indian Institute of Science, Bangalore. It was all those days ruled by British. So 1904, who discovered neon, krypton and xenon and received a Nobel Prize in 1904, and he was the first director of Indian Institute of Science, Bangalore. Okay, so I am closing it there. If you have any questions, further questions, I will be able to answer.
Sir, in the beginning you had said that we mimic the biological process which happen inside an organism. Why do we do that?
Which one? Why do we?
why do we do that? Like oxygen binding reversibly, you said that we try to do that in.
Why do we do that? Because metalloproteins and metalloenzymes very complex to understand, very difficult to crystallize and know their structure. So through spectroscopy, if you can design molecule, finally at the end ultimately you want to have an artificial blood, for example. So whether you have made a similar compound or not, how do we know? And every time you cannot understand these proteins and the enzymes which are very complex in structure. So and basically this structure and function happens only at the site where metal is present. So if you can mimic and duplicate the property of that active site, maybe we know that how it functions, how it does, and what happens, how it is reversibly binding or not. Through spectroscopy, if it is matching with the same thing that what is observed in biomolecules, that means okay, maybe this is the reason, this is what is happening. For example, nobody knew that iron transfers one electron to oxygen and becomes a superoxide. Nobody knew. People always thought iron remains as a +2. There is some weak coordination, oxygen gets reversibly binds. That's what people thought in the beginning. Now through many different such kinds of simple molecule design, that people have understood this is what is happening. Iron gives one electron to oxygen, and then oxygen in turn gives one electron back to iron, and that's how it binds reversibly in the case of hemoglobin. So this gives us some idea how these molecules could be functioning. Otherwise, all the time it is very difficult to study the protein structure and function. Functions we know, but how they are doing, you want to know in detail.
Sir, you have talked about methyl mercury.
Huh?
Methyl mercury, sir.
Metal?
Methyl mercury.
Metal.
Is that a coordination compound, sir?
Is a coordination compound.
Sir, how is methyl a ligand here if it doesn't have a lone pair?
Metal has mercury. That is what you are going to study, what is called organometallic chemistry, where the organic molecule with a metal forms when the metal is the zero oxidation state. Okay, those kinds of compounds are called organometallic compounds. Okay, so mercury is zero oxidation state, and methyl mercury bond is very strong. It will form CH3 minus will interact with the mercury 0. Yeah, it's not necessary that it has to be this. The compounds containing metal in the +1, +2, +3 or 7 and the rest are all called coordination compounds. So those contain metal in the zero oxidation state, they are called organometallic compounds. So they are non-classical coordination compounds. These are all the classical coordination compounds. No other questions? No? Okay, thank you. Yeah.
Sir, on behalf of all the RSIC students, I express my heartfelt gratitude for gracing us with this lecture today. The understanding on the coordination chemistry and how it was applied in biology was truly enlightening to us. Thank you so much for your presence and for this.
That is only after your 12th.
Yeah.
If you understand some of the things, now I am assuming that you will have interest why we want to really study this coordination chemistry, because it has a lot of applications. So after the course is over, now if you recall and now you are paying more interest because there is an interest application, then it is useful.
Yes, sir. It will truly resonate with us, and we'll assure you that we will explore more on this.
Oh, it's very nice.
Thank you so much.
Wish you all the best. Yeah.